On today's FSL podcast, we are going to be exploring the topic of gene editing. But wait, let's back up. Firstly, what is a gene? Genes contain information the body needs to protect itself from foreign invaders and rebuild itself from damage. This information comes in the form of specific sequences of base pairs. Essentially, each base pair can be represented by a letter in a sentence. A certain order of letters creates a readable sentence that instructs the body on how to act in a certain situation. In summary, genes store the information our body needs, and this information is read from specific sequences of base pairs. So, to answer the ultimate question, what is gene editing? Gene editing is the act of altering these sequences of base pairs. To do this, scientists use a fascinating technology known as clustered regularly interspaced short palindromic repeats associated protein 9, or CRISPR-Cas9. This technology involves two parts, a guide RNA and Cas9. Once scientists have identified what sequence they want to edit, they program the guide RNA to look for this specific sequence in the gene. Then, once found, the Cas9 cuts out the sequence and replaces it with a sequence of base pairs that the scientists have already curated. And that's it. The instructions that cell follows has now been altered. This technology, CRISPR-Cas9, and gene editing in general, has huge potential world-changing applications. Currently, its biggest applications have been seen in agriculture, where 20 to 40% of crops are lost due to climate change, diseases, or pests. CRISPR-Cas9 has already allowed scientists to engineer plants with better resistances to these hazards, dramatically increasing the plant's survival rate. CRISPR-Cas9 is an emerging technology that must pass many ethical barriers before it can be used on humans, but it has already been proven as a powerful tool when used in the agriculture space, with the potential to permanently change how we approach genetics and biology. And that's gene editing for today's FSL podcast.